Hello and welcome back to another Dr-Tutor video. Today we're going to be covering the A-level biology topic of biological membranes. This video has been catered more specifically to the OCR syllabus, but has been adapted slightly so that it should be applicable to all major specifications. It's important to understand the roles of membranes, both within and at the surface of cells. So at the surface of cells, membranes play a crucial role in separating the contents of a cell from the environment. They're also partially permeable, so they can control what enters and exits cells. They also play a key role in cell signaling, as they can release signaling molecules and they have receptors on their surface. They can also act as the site of some metabolic and chemical reactions, as they contain the enzymes involved, and they, as mentioned, have receptor sites for hormones, and will have self-antigens to assure it's recognized by the immune system, that the cell itself is recognized by the immune system. Within cells, they play a crucial role in compartmentalization, assuring that organelles can be separated from each other and the cell itself, and they can play a role in forming vesicles for exocytosis, as well as endocytosis. Within organelles, such as mitochondria and chloroplasts, they form a folded membrane and can play a role in increasing surface area and assuring. Moving on, when we refer to cell membranes, we often hear of the fluid Mosaic model, which is describing a cell membrane structure. So the main base of the membrane is the phospholipid bilayer, with numerous transmembrane proteins such as carrier and channel proteins. This accounts for the mosaic aspect of the name. The fluidity is because the phospholipids can move around and be fluid within the membrane as opposed to being fixed in position. It's important to note that phospholipids are arranged such that the hydrophobic tails are inward facing and the hydrophilic heads are outward facing towards the kind of watery cell contents and outside of the cell. The proteins allow ions to pass through via facilitated diffusion, and the carrier proteins undergo conformational changes to enable some molecules in the cell to enter and exit the cell. Some proteins can be attached to carrier proteins or receptor sites to form glycoproteins for receptor sites. It's also important to have an understanding of the role of cholesterol in cell membranes. So cholesterol increases the fluidity of membranes, stopping them from becoming too rigid at low temperatures and thus allowing cells to survive at these lower temperatures. This occurs because cholesterol stops the phospholipid tails packing too closely together. Interaction between cholesterol and phospholipid tails also stabilizes the cell membranes at higher temperatures by stopping the membrane from becoming essentially too fluid. So cholesterol molecules bind to the hydrophobic tails of phospholipids and stabilize them, and thus they pack more closely together, which increases the rigidity of the membrane. Based on your specification, it may be worth noting there's also a glycocalyx which lies on the outside of the cell membrane, and this is composed of glycolipids and glycoproteins, which are lipids and proteins respectively attached to carbohydrate chains. Not all membranes are identical, so neurons, for example, are different, and many specialized cells will have adaptations for their given functions. So the fluid mosaic model of membranes was outlined in 1972, and its primary function is just to explain how biological molecules are arranged to form a cell membrane. It also helps us explain the passive and active movements between cells and their surroundings and how cells interact with each other. There are a number of factors that affect membrane permeability, and these are seen on screen. Now we're going to go through these somewhat briefly, as there are tons of notes on these topics and all topics in the A-level chemistry and biology syllabus available on dr-tutor.co.uk. But going into each of these factors in turn. Temperature. As temperature increases, the molecules gain kinetic energy, so the rate of diffusion increases and vice versa as temperature decreases. So it's important to know that proteins and phospholipids are both affected by temperature. So as temperature increases, the phospholipids in the bilayer become more fluid. So this increased fluidity reduces the effectiveness of the cell membrane as a barrier to polar molecules, meaning some polar molecules will be able to pass through. At higher temperatures, any diffusion taking place through the cell membrane will also occur at a higher speed due to increased kinetic energy. It's also important that we understand that changes in membrane fluidity are reversible, such that if temperature decreases, the lipids will return to their normal levels of fluidity. The phospholipids will return to their normal levels of fluidity. However, at certain temperatures, which are often around 40 degrees centigrade, many proteins begin to denature, and this can be true of the cell membrane to an extent as well. This disrupts the membrane structure, meaning it no longer forms an effective barrier, and as a result, substances can pass freely through the disrupted membrane, and denaturing is an irreversible process. Moving on to diffusion distance, in essence, a thicker membrane means a slower diffusion rate. With regard to surface area, the larger surface area means more diffusion can take place. We can think back to microvilli, for example, where we attempt to increase surface area as much as possible to increase the area across which substances can diffuse. The size of the diffusing molecule is important, as smaller molecules diffuse more rapidly, and the size of the concentration gradient is also important. So a steeper concentration gradient will lead to a faster rate of diffusion. In some cases, organic solvents can increase cell membrane permeability as they dissolve the phospholipids in the bilayer, causing the membrane to lose its structure, but this is not necessarily a factor that affects membrane permeability in the traditional sense. Now we have the various ways molecules can move across the cell membrane. So the first and perhaps most well known is simple diffusion. This is the net passive movement of molecules from an area of high concentration of that molecule to an area of low concentration down their concentration gradient across a partially permeable membrane, and this is only applicable to small non-polar molecules or lipid-soluble molecules, as these dissolve in the bilayer essentially. Next is facilitated diffusion, and this is the net passive movement of molecules from an area of high concentration of that molecule to an area of low concentration down their concentration gradient across a partially permeable membrane via protein channels or carrier proteins. This is applicable to small polar molecules such as ions or large necessary molecules such as glucose, say, which can bind to transmembrane carrier proteins, and these carrier proteins will be specific to the molecule that they transport. There are also channel proteins, and these are essentially water-filled pores which allow substances to diffuse through them, and specifically charged substances to diffuse through them, such as ions. The diffusion of these ions does not occur freely, and most channel proteins are gated, meaning that part of the channel protein on the inside surface of the membrane can essentially move in order to close or open the pore, much like a gate. As such, the channel protein can control the exchange of ions to an extent. Carrier proteins, unlike channel proteins, which have more of a fixed shape, carrier proteins can switch between two shapes. This causes the binding site of the carrier protein to be open to one side, the open side of the membrane first, and then open to the other side of the membrane, essentially changing shape and allowing a molecule to pass through it, if that makes sense. The direction of movement of molecules diffusing across the membrane depends on their relative concentration on each side of the membrane. Now we can move on to osmosis. Osmosis is the net passive movement or diffusion of molecules from an area of high water potential to an area of low water potential down their water potential gradients across a partially permeable membrane. Osmosis is only applicable to water. We cover osmosis in a bit more detail shortly in a few slides. So next we have active transport. This is the movement of substances or molecules from an area of low concentration of that molecule to an area of high concentration of that molecule against their concentration gradient, or up their concentration gradient, using ATP energy and protein carriers. So the energy is provided by the hydrolysis of ATP, which is produced through respiration, and this occurs as cells and organelles may require more of a particular ion or molecule than they could get through diffusion or facilitated diffusion alone. An example of this would be in root hair cells for the absorption of ions. Active transport uses carrier proteins which have specific sites to combine with only certain solute molecules to allow the hydrolysis of ATP to provide the energy for the carrier to undergo a conformational change, or a change in shape, to carry an ion from one side of the cell to the other. Bulk transport is an active process that requires ATP energy and is for large molecules or particles that cannot fit through the membrane, and thus a pump is essentially used to transport molecules from one side of the membrane to the other. So moving on to endocytosis, this is the process by which large particles are brought into the cell when they can't pass through the plasma membrane, and instead the membrane surrounds them and encloses them in a vesicle, and this vesicle is then brought into the cell. Penocytosis is part of endocytosis, and this is the bulk intake of liquids. We then have exocytosis, which is the method to remove large molecules from the cell, whereby a vesicle containing these molecules will fuse with the plasma membrane, and an example of this would be, say, with neurotransmitters. So going into osmosis in a little bit more detail. As we mentioned, osmosis is the net passive movement of molecules from an area of high water potential to an area of low water potential down their water potential gradients across a partially permeable membrane. This, as we mentioned, is applicable only to water. So when more solute is added to a solution, the relative number of water molecules of the solution is changed, and if they dissociate, they exert more of an effect on the relative number of water molecules. Water potential is essentially the tendency of water molecules to diffuse from one area to another. Pure water has the highest water potential, which is 0. So when solute is added, they lower the water potential. And it's really important to know that the highest water potential is 0 kPa and becomes more negative. So osmosis can have a variety of different effects on cells. So in animals, if the water potential of the cell is far lower and lots of water enters the cell will essentially swell and burst. This breaks the plasma cell membrane, and we call this the process of cytolysis, and this occurs in hypertonic conditions. In plants, if lots of water osmoses into the cell will prevent bursting, but the cell membrane will swell and exert more pressure on the cell wall, making the cell turgid. In animals, if cells are in a solution with lower water potential and water leaves the cell will be cronated, as the cell essentially shrivels. In plants, if cells are in lower water potential solutions, the cell membrane shrinks and pulls away from the cellular cell wall, and the cell is described as plasmalized. This can make the leaf and the cell flaccid, and this will occur in hypertonic conditions. So that's hypotonic conditions for cytolysis versus hypertonic conditions. So here we see just a series of images that illustrate what cells look like in each of the stages we mentioned on the previous slide. Feel free to pause and kind of understand what's going on in each stage, and why each cell looks the way it does. So to some extent that brings us to the end of the topic, but this is not everything included in the topic and is definitely not in full detail. Feel free to head over to www.dr-tutor.co.uk for more information, and you can also book one-to-one tuition sessions where we can cover all of the topics in A-level chemistry and biology or GCSE, and go over them in far more detail, and I can answer your subject-specific questions, and we'll also go over exam technique and how to answer these questions, because that is a really tricky part of the A-level in general. And if you're not interested in booking one-to-one tuition sessions, there are also free notes and interview preparation for prospective medical students, and those are included, as mentioned, some of them completely free, and you can head over there, and that covers the topic in full detail, especially for OCR for biology. overall, thank you for listening, and I hope you join in future sessions to cover more A-level biology and chemistry topics. So see you next week.